Okay, so I'm going to go over with you some Unit 2 and 3 concepts to help you prepare for our exam this week, next week. So just keep in mind, if you're in Period 7 class, the non-blended class, on Monday we'll have our Unit 2 and 3 free response question. And then Tuesday we'll have our 65 multiple choice portion of the exam. Both both the FRQ and the multiple choice incorporate information from both chapters 2 and 3. If you're my blended class, on Wednesday we will take a different 65 multiple choice question exam, again on Units 2 and 3. Your job with the FRQ will be to complete it, the one I posted online, by Wednesday's class. Sorry, by midnight. Okay, so some of the key things you want to make sure you're aware of, there's a lot of definitions here you want to make sure you're able to keep straight. When we're talking about hindsight bias, that's the tendency to believe that we knew it all along, even if we actually didn't. It just seems like it makes sense, like, oh yeah, after the fact I knew that. There's also, okay, so then there is overconfidence, thinking we tend to know more than we actually do. We did the one activity in class having to deal with the 98% accuracy of the length of rivers and things like that, and we saw that we are all overconfident. Scientific method, definitely expect to be able to need to explain or understand kind of the process of the scientific method and why that's in place. Definitely know with a hypothesis, I believe there's a definitional question here. Just understand with a hypothesis that you need to make sure that it's brief, and a key thing is you must make sure you have operational definitions so that we can repeat it over and over and over again with different researchers but the same exact kind of experiment. If you remember operational definitions, we did the example where we had all the pictures of people smiling, but different people had different interpretations of what smiling was. So we needed to make sure we described that, operationalized that in real life terms, so that way someone could repeat that same experiment. Okay, descriptive research is definitely a key idea you're going to need to be able to know for this exam. Definitely remember the survey method, you know, the good and the bad. Remember you have that chart in your notes, or you may have typed into it, and that kind of goes through the pros and cons of what you need to know about different kind of research methods. With the survey, it could be oral interviews, it could be questionnaires, and it's really, really easy to get a random sample of respondents. Stuff you must know for the AP exam and then also for my exam is that when we're talking about a population, that's just the whole group I want to study. So if I wanted to study the behavior of kindergartners, my population would be kindergartners. And then the next step after you get your population is you want to pull from that population, because I don't want to talk to every single kindergartner in the whole world, right? So I need to make sure that every kindergartner has an even chance, an equal chance of being selected. So you could use a random name generator, you could use a random number generator, just to make sure that everyone has a chance of being selected. Once you have your sample, you then need to split it into your experimental and your control group, and that requires random assignment. So basically, everyone needs to have an equal shot at being in the experimental group or being in the control group. I couldn't put all boy kindergartners in one group and all girl kindergartners in another group, because that's a confounding variable, something that could mess with the results. Keep in mind, obviously, the bad about the survey method is people could lie. Case study, think of Phineas Gage. Think of cases that you could not reenact or replicate, so you would only study one particular person or situation in depth. Naturalistic observation is when you're observing something, behavior possibly in its natural environment. If you want, that's a lot of times when you're observing animals in the wild, or you might be looking at people through that two-way mirror type thing. Experimental research then is quantitative research. This is the only way that you can prove cause and effect relationships. Remember, correlation does not equal causation. To prove causation, you must have experimentation involved. Correlational research, remember, that just detects relationship between two or more variables. So we talked that there's a positive correlation between ice cream sales and murder rates, but we know that it's just because we have that third variable of summer and hot weather. So we know that ice cream doesn't cause murder or murder isn't attractive to ice cream eaters or something like that. There's just a positive relationship there. So remember, you have positive or negative correlation. You need to understand the difference between the two and also how to read those correlation coefficients. If it's a positive correlation, remember that means the variables go in the same direction with one another. You can do the hand thing. Positive, they like each other, they want to move in the same direction. So for example, studying and grades has a positive correlation. As one goes up, so does the other. As one goes down, so does the other. On the other hand, the negative correlation means the variables move in opposite directions. They're negative, they don't like each other, so they want to move away from one another. So heroin use or drug use likely has a negative correlation with grades because as heroin use goes up, grades go down. Heroin use goes down, grades go up. We must know what a correlation coefficient is, but most importantly, how to read that correlation coefficient. Remember, it's going to have either a positive or negative symbol in front of it, or if it doesn't have a symbol, that means it's positive. It's implied that it's positive. If it's positive, that means it's a positive correlation. If it's negative, that means it's a negative correlation. The strongest negative correlation you can have is a -1. The strongest positive correlation you can have is a +1. The closer you get to 0, that just shows that there literally is no relationship at all. So right here, for example, this R positive 0.37, it shows us a little bit of relationship, not super strong, and it's obviously in the positive direction. I know I do have on our test some graphs that you need to read. I know there was some discussion in, I think, my period 7 class about how to read this, but also we looked at this in period 2. Perfect positive correlation is going to have the perfect slope of 1, so it's going to be sloping upwards. If you have no relationship, as you can see here, you're going to have your dots and your scatter plot all over the place because there is no relationship. If you have perfect negative correlation, we're going to have that slope of -1. Illusory correlations, these are correlations which we perceive relationships, but there actually is none at all. I think that's a definitional question on our exam. Okay, I know one of the FRQs has you kind of talking about an experiment or designing an experiment, so I want you to be familiar with those terms for sure. An independent variable, that is what the researcher is manipulating in the experiment, and you're hoping that the independent variable somehow brings about change, which will then be reflected in the dependent variable. So in the dependent variable, that's what we're measuring. We're trying to see what kind of impact it's going to have and be able to measure that. As I said earlier, you need to make sure you're aware of confounding variables, because if I was running an experiment or a study on that smoking causes heart issues, I got a lot of other stuff to consider. I got, what if this guy's in my experiment has heart issues, but it's not because he smokes, it's because he sits on the couch and eats food and watches TV all day. Sampling, we just talked about that. Random assignment as well. Okay. Experimenter bias, make sure you are aware of the difference between double-blind procedure and just a blind procedure. In a blind procedure, so right here, let's say this guy he's explaining the experiment. He's explaining, okay, some of you are going to get this drug, some of you are not going to get the drug, but he's not going to tell them which of them is getting the drug and which isn't. The control group would then get some kind of sugar pill, so that would be a blind procedure. A double-blind procedure is used, and the researcher wouldn't even know who's getting what, so that way the results are not biased in any way. Okay. Statistical significance, I believe that there is a definitional question on the exam. This is a p-value, okay? So this is the likelihood that a result of an experiment is caused by chance. It cannot be greater than 5%. So researchers accept that, okay, there's probably a 5% chance here or less that some kind of our result was due to chance, but researchers will not accept anything greater than 5%. Replication, just understand that you need to be able to repeat experiments to get conclusive results. Statistics-wise, you definitely, there are some questions asking you to compute the mean, median, and mode. Those are all measures of central tendency. It doesn't really tell us a lot of information about the extreme outliers, the extreme scores. In a normal distribution, we talked about this a little bit, and I believe I drew this on the board. In a normal distribution in a perfect bell curve, the mean, median, and mode are all exactly the same. Distributions then, because we have to account for some of those outliers, we do have certain skews that may occur. We have positively skewed distributions where an outlier is pulling those that data outwards positively. Think of it like a number line. And then a negative skew means there's some outlier that's pulling it out negatively. Okay, and I just explained that right here. Feel free to look at this PowerPoint as well. Measures of variability then, to kind of account for those outliers. We have range, that's the distance from the highest to the lowest scores. And then standard deviation is the variance of scores around the mean. Researchers want a smaller standard deviation because they want those scores to be clustered together closest to the mean. Okay. There are percentages you need to know for the exam. So if you remember, all people fall within, 68% of people fall within one standard deviation of the mean in a normal curve. Okay, this right here is the Wechsler Intelligence Score Scale. No matter what information, what data you're looking at, if you have a normal distribution within one standard deviation, you always have 68% of the population. Within two standard deviations then, as you can see from here to here, you have 96% of the population, okay? And then we have kind of 2%, 2%, and then at 0.1%. I know it doesn't equal exactly 100, but it's as close as we're going to get. Okay, now I'm going to quickly transition into bio stuff. So the neural and hormonal systems. Okay. So we need to make sure we know the difference between the three different types of neurons. We have sensory neurons, motor neurons, and interneurons. Sensory neurons, also called afferent neurons. Remember, it takes information from the senses. I know you can't see my hands right now, but it takes it in. So sensory neurons are afferent neurons. So afferent in, afferent in, afferent in. That takes information from our senses and brings it to our brain. There are millions of sensory neurons. There are also millions of motor neurons. What motor neurons do is they take information from the brain to the rest of the body. So those are also called efferent neurons. So afferent in, efferent out. Afferent in, efferent out. Try to keep that straight in your mind. Interneurons, are billions of them, and they take messages from sensory neurons to other parts of the brain or to motor neurons. Again, we have billions of these interneurons. Neuron structure, I know we labeled the neuron a lot in class, but you also want to make sure you understand the function of each part of the neuron, so I'd highly recommend reviewing that, possibly making some note cards, okay? We know that the dendrites receive information from other cells from the previous neuron. We know the cell body, that's kind of where all the life center is in that neuron. The axon, I call it the tubey thingy, but basically it just passes messages along the body. We have the myelin sheath, that's like the insulation you have in your attic to kind of insulate, keep that electrical impulse going. And then we reach the terminal branches at the end of the neuron where they have vesicle sacs with those neurotransmitters located within them. Also remember, the neural impulse can only go in one direction, from the dendrites to the terminal branches. It cannot come back up. Okay. Remember how a neuron fires is going to be important as well. It's an electrochemical process. We know inside it's an electrical process. In the synapses, when we're dealing with the neurotransmitters, that is a chemical process. Please keep in mind, it is that all or none response. A neuron doesn't fire just a little bit. It either fires or it doesn't fire, okay? So it's just like a gun. Steps of the action potential, right? I kind of mentioned that briefly, but basically the dendrites are going to receive the neurotransmitters. Once the receptor sites reach their threshold, they're like, okay, I'm closing my portal, I'm ready to go, I'm ready to fire my action potential. And then we have that whole process where you're going to open like a little door, ports in the axon. It's going to let the positive ions in, which is going to mix with the negative ions that are already inside the axon. And then the mixing of those ions is going to cause an electrical charge that will, like I said in class, kind of open the next door, allow the next situation to happen, and that process continues down all the way to the axon terminal. And the terminal buttons turn that electrical charge back into the neurotransmitters, that chemical process that's going to shoot across the synapse.